Hello, my name is Natalia Padilla. I'm an incoming junior at UC Berkeley, and this summer I worked under the mentorship of Dr. Zhao, in which I researched PKC epsilon activation and the pathogenesis of non-alcoholic fatty liver disease. Our main research question this summer was, is the activation of PKC epsilon in non-alcoholic fatty liver disease in transgenic humanized resistin mice induced by a high-fat diet or resistin? Understanding the pathogenesis of non-alcoholic fatty liver disease is important because of its prevalence in developed nations, in which it falls between 20 to 30%. PKC epsilon is a type of enzyme that is activated by lipids and diaglycerol, which phosphorylates the insulin receptor substrate 1. We collected liver tissue samples from 12 transgenic humanized resistin mice with NAFLD that were either fed normal chow, had resistin overexpressed, or were fed a high-fat diet, or had both resistin overexpressed and were also fed a high-fat diet. Then we homogenized the tissue, first using a scalpel and then a pistol. Then we isolated the total protein of our samples by collecting the supernatant in pellets. Then we performed a PKCe assay to find the total protein concentration of our samples in order to run CDS page. Lastly, we used immunodetection to determine the presence of B-actin, the total PKC epsilon, and the total activated PKC epsilon in our mouse liver tissue samples. Although our results were inconclusive, there was higher total PKC epsilon and total activated PKC epsilon in our high-fat diet transgenic humanized resistin mouse group. My final questions are, why did humanized resistin transgenic mice with resistin overexpressing on a normal chow diet induce lower PKC epsilon activation in comparison to a high-fat diet? And why is there less total protein concentration in humanized resistin transgenic mice liver tissue with a higher fat composition? How does this affect the function of the liver?